University. He also pursued a postdoctoral fellowship at the Hammersmith Hospital in London, where he researched movement disorders and functional brain imaging. And he also contributed to brain imaging methods, and most notably, he developed a novel method for measuring in vivo dopamine release in the human brain using positron emission tomography. And he then returned to the Montreal Neurological Institute, where his research currently combines two components. They try to understand Parkinson's disease and identifying the neural mechanisms that support motivated decision-making with applications to addiction and obesity. And what is really fascinating, I think, is that he manages to combine functional and anatomical brain imaging in human subjects with genomics, psychological, economical, and cognitive measures to understand how a brain becomes vulnerable to disease and maladaptive behaviors, and thereby really manages to bring together seemingly disparate symptoms and diseases in an overarching framework. And I'm very excited that today he will share with us his research about obesity and the brain and bidirectional influences. And I will stop here and hand over to Alain, and thank you for being with us today and share your work and discussing it. So the stage is yours.
Thank you very much for this invitation. It's a great pleasure to participate in this really outstanding seminar series. So today I will talk about data showing that obesity and brain influence each other, and try to understand the direction and the causality of these relationships. So first I would like to thank the people who have done the work that I'm going to present today. In particular, on the left are the postdocs and students, Uku Vainik, Celine Nesseliller, Isabel Garcia, Philip Morris, who did most of the work that I'm going to present today, as well as some of our other collaborators and funding bodies. So you know that the gut and the brain talk to each other, and the main reason they talk to each other is to control food intake.
It seems that we have lost him for a second.
Okay. Am I there?
Yes, you reappeared. I think you had a connection issue and we lost you for a second.
The crowdcast seems to keep going on and off.
Is it your internet connection or is it Crowdcast? Because otherwise we can also switch over to Zoom if that works better.
I think my internet connection is fine.
Okay, now actually I can hear you very well. Beforehand there was some background noise.
Okay, I don't understand. So let's
try it once more. If it happens again, we will just switch to Zoom. I will share the link.
Okay. Okay, so as I said, often when you look at work on energy homeostasis, it seems that people think it stops about at the red line that I drew, namely at the hypothalamus. So there are multiple connections between the gut and the adipose tissue and all of the organs and the brainstem and the hypothalamus. And the question is, does the control of energy balance stop there, or does it involve higher order cognitive systems in the brain? And that's a question I'll try to answer. Not sure that I have the answer, but at least I'll look at the evidence. So we know that homeostasis and allostasis tightly regulate food intake. However, we also know that there's been an increase in obesity and BMI in the last 50 years, and it's clear that the main driver of this is the low cost of food, which doesn't make sense from the standpoint of homeostasis. It's not clear why cheaper food would lead to overconsumption. And I'm going to look at evidence that body weight in humans is actually under the control of executive function and prefrontal systems, as well as homeostatic and gut peptide mechanisms. And one reason for this might be an economic thrifty hypothesis where it is optimum to eat more when the food is cheap or abundant. And if that's the case, then abundance of food would signal overeating, perhaps by reducing self-control. So humans are omnivores. We can derive nutrients from any environment. That's a tremendous advantage. It means we can live everywhere on Earth. However, it also means that you must learn about the constituents of food, of novel foods that you consume. And so unlike, say, pandas and cows that eat only one thing, humans have to learn about the foods that they eat. And this brings in the central nervous system. This shows in blue the increase in the incidence of obesity since the 1980s, and you all know this curve. But in red, it shows total credit market debt, which is a measure of consumption in society. So it seems that as we consume more of everything, we also consume more food, and this has led people to take an economics approach to the obesity pandemic. So recently, many groups have, so one of the aspects of the change in the food environment over the last 50 years is the ability to process food and to change its characteristics, in particular to change its caloric density. And there are many studies that show that consumption of ultra-processed foods is linked to cardiovascular and cerebrovascular mortality. These are two different studies, the Framingham study and a study in France with over 100,000 individuals. And the more you consume of ultra-processed foods, the more you have heart and cerebrovascular disease. And obviously, this is via obesity. And so the more you consume ultra-processed foods, the more obese you are. Why is that? Well, so this study by Kevin Hall tries to answer the question. Here what they did is they brought people into an inpatient unit where the food that they could consume was completely determined by the investigators. People were allowed to eat ad lib. So for 2 weeks they had ultra-processed foods, and for 2 weeks they had an unprocessed diet, that is mostly raw, mostly natural foods. And they were still allowed to consume whatever they wanted in both cases. And what you can see is that on the ultra-processed diet, there was an increase in body mass index, as well as an increase in consumption of calories. So ultra-processed foods, in other words, are designed to be overconsumed. So as I said, they're engineered for consumption and probably actually for palatability, and therefore are more likely to be wanted, purchased, and eaten. Food companies can manipulate foods almost infinitely, but the thing they can manipulate the most to get us to consume more is caloric density. And again, this goes against homeostasis, but people are sensitive to the cost of food, so-called price elasticity. This has been demonstrated even for things like milk, which is a staple. And it's estimated that about half of all people are environment sensitive. In other words, they will gain weight when food is cheap or abundant. And people have called this the thrifty phenotype. So this sounds very much like something that is not purely homeostatic, but more allostatic, meaning a behavior that attempts to predict the future and attempts to predict future needs. So in this setting, allostasis would balance needs, such as your current level of energy balance, and opportunities, such as the current level of food availability or the price of food. And this diagram looks very much like an economic diagram where costs and needs both influence decisions. So we've been using this paradigm from the neuroeconomics literature for some time now. And here what we do is we ask people to bid on the right to consume food. So we show them repeated images of familiar foods and ask them to bid a certain amount of money to determine whether they will be allowed to consume the food at the end of the experiment. And that bid amount is a measure of wanting. So this is a technique that's been used in economics for many, many years. And Antonio Rangel and Hilke Plastman combined this technique with functional MRI. So here what we do is we have people bid for various food items while they're being imaged with fMRI. fMRI basically just measures brain activity or neuronal activity. And then we look for brain areas that encode or that correlate with the BID amount. So in other words, correlate with wanting. And you see that there are many regions in the brain where brain activity, when you're evaluating these foods, correlates with wanting. In particular, the nucleus accumbens bilaterally, the midbrain, where dopamine neurons are present, the anterior cingulate cortex, the ventromedial prefrontal cortex, the orbitofrontal cortex, dorsolateral prefrontal cortex. So all of these brain areas implicated in decision-making and evaluation seem to track the cost, the wanting of these items. And in particular, this area has been very interesting, this ventromedial prefrontal area, which Plasmid et al. Showed tracks the wanting or the BID amount of items, not just food items, but almost any kinds of items. So this is an area which appears to be computing the value, the subjective value of an item. We showed with Deborah Tang that in this study, the BID amount, the wanting, correlated with caloric density. And in fact, it correlated with the true caloric density of the food items, although it did not correlate with the estimated calories. When we asked people to estimate the calories in foods, they were very bad at it. But that caloric density seemed to correlate with how much they wanted or liked or enjoyed the foods. And then remember this vmPFC area, which tracks wanting. When we correlated the true caloric density of the food items, we found that it also tracked this area, this value. So this evaluation area seems to be activated in proportion to the caloric density of the food items. And we also already know that this area is involved in motivation and wanting. So we desire food items that are calorically dense more. And Drunovsky and colleagues have shown that caloric density was inversely proportional to the cost per calorie. And this is the result of processing. So if you have the ability to make calorically dense foods cheap, and we know that most humans want calorically dense foods, then in an economic system,
I think we lost him again, and he seems to be reconnecting. I think we lost you once more. It said you were reconnecting, but now I can see you again.
Okay.
And I can also hear you very well.
Okay, let me stop. Let me keep going. Yeah, I don't know what's happening. Should I keep going?
I think so. It seems to be the internet connection.
Let me try something. How is it now?
I think it's the same for me.
Okay, but you can hear?
Yes, I can hear you now very well.
Okay. Okay, so what I was saying here is that there is an adaptive effect of over-consuming when food is abundant, because doing so will lead to increased fertility. So genes that are implicated in this behavior, that is in increasing consumption when food is cheap, will be under selective pressure. We know that body mass index is heritable, and interestingly, this GWAS study, already 8 years old, there are now bigger ones available, but identified about 100 alleles that contribute to body mass index. What this suggests is that BMI is polygenic, and in fact it's estimated that as many as 2,000 different alleles might contribute to BMI. Another interesting fact is that your number of risk alleles predicts your BMI. So there's a linear correlation between the number of BMI risk alleles that you have and your body mass index. And this is the distribution in the population of these risk alleles. So what are these people? These people over here with the high risk alleles, why is that? Why are they overweight? It could be that they have greater appetite. It could also be that they have greater tendency to overeat in response to food abundance. And those things might be related in themselves. This study, I won't go into too much detail. You can read it if you're interested. But what it suggests is that those genes that are implicated in body mass index or related features are under strong selective pressure. This is by comparing the relation of the minor and the major allele at each point. So the story so far is that overeating in response to signal of abundance appears to be genetic and appears to be selected under selective pressure. And what is probably happening is that people are matching their level of consumption to the environment. And to do that would implicate higher level cognitive systems, because you have to take into account multiple sources of information about food and the food environment. But what is the endophenotype that leads people to overconsume in response to abundance? So this is a paper by my ex-postdoc Uku Vainik, who looked at all the neurocognitive measures linked to obesity. And he found that there were many, so each of these is a different cognitive test, and the ones that are in green or yellow show an association with BMI in most studies. And what you can see is that in the area of executive function, especially in the area of inhibitory control, there are very strong and consistent associations. For example, the Stroop task and the stop signal task. There are also strong associations with food motivation, tasks of how motivated you are by the presence of appetitive foods. So this led to this concept of uncontrolled eating as a trait, as a phenotype, in which both sensitivity to food reward and self-regulation interact to produce overeating in response to food abundance, and therefore leading to obesity. Now there are many ways to measure these reward sensitivity and self-regulation facets. One is by questionnaires, such as the BIG-5 personality test, or by lab tests. And then these may themselves be associated with structural or functional MRI features. There are other extrinsic factors that may modulate uncontrolled eating, such as stress. And then there are various other factors, such as socioeconomic status, physical activity, that may modulate the relationship. So the idea is that this uncontrolled eating is a trait, it's a phenotype, it's heritable, it's genetically determined, and what it does is promote overconsumption when food is abundant. Uku and Ashley Mason and Alyssa Eppel and myself developed a questionnaire for uncontrolled eating. It's called the RED scale, the Reward Eating Drive scale, and it's derived from multiple other food-related questionnaires. And you can compute each individual's latent RED score, which we think is a measure of uncontrolled eating. And you see that in this undergraduate sample of 2,000 students at McGill, that the uncontrolled eating latent score was quite strongly associated with BMI. And then we did a structural equation model to try to relate various cognitive measures to uncontrolled eating, and found that there are three traits that relate: reward sensitivity, lack of self-control, and stress reactivity. So these three interact together and with each other to cause this uncontrolled eating phenotype, which is itself associated with obesity. And then if you're interested, you can look at the paper. We looked in a small subsample of 75 people, which is probably inadequate, but we looked at cortical morphology and its relation to stress, reactivity, reward sensitivity, and lack of self-control. So now we come with this idea that appetitive response to food abundance is partly under the control of higher level cognitive functions and under the control of, therefore, cortical systems, not just under the control of the hypothalamus and hunger. And this meta-analysis looked at self-regulation of appetite in fMRI, and what we found is that when people are asked to regulate their desire for tasty foods, we have activation of the dorsolateral prefrontal cortex, ventrolateral prefrontal cortex, and anterior cingulate cortex, a group of areas that are consistently implicated in executive function. And remember that I said executive function was related to BMI, again in multiple studies. So we did this study in people who underwent a weight loss regimen. So these are people who are somewhat obese, BMI in the 30s, otherwise healthy, who went on a calorie restriction regimen of about 800 to 1,000 calories a day, so quite strict. And they did this for 3 months, and we imaged them before, at 1 month, and at 3 months. And we did the same kind of study that I've shown before, where we show food pictures and ask them to rate how much they want them. So we found, first of all, that voluntary calorie restriction is effective. You can see that there's a reduction in BMI. Baseline is in purple, blue is 1 month, green is 3 months. You can also see that there's a reduction in leptin. Leptin is a hormone that is basically an indicator of the state of energy balance. It's secreted by the adipose tissue. So leptin, as expected, goes down when you lose weight. Another hormone, which is kind of the anti-leptin, is ghrelin. Ghrelin increases hunger and increases appetite, and you can see that ghrelin goes up as well after weight loss, possibly promoting greater consumption. So then we were interested, what in the brain predicts successful weight loss? So we looked at brain areas where activity, while evaluating the food pictures, predicted weight loss. And you can see that it's these same areas that I showed before, these bilateral, lateral prefrontal areas and anterior cingulate areas. So this really very clear executive network, the greater your ability to activate that executive self-regulation system when looking at appetizing foods, the greater your ability to lose weight voluntarily. So this also supports the idea that cortical high-level cognitive systems are implicated in energy homeostasis. And then we looked at signals associated with successful weight loss, and what you can see, this ventromedial prefrontal area, which is implicated in valuation, there's a reduction at 1 month, and it starts to come back up at 3 months. Meanwhile, this dorsolateral prefrontal area, implicated in self-regulation, increases at 1 month and decreases at 3 months. And this is related to successful weight loss. not only do these areas show this effect, but they are functionally connected. And the degree of functional connectivity between lateral prefrontal cortex, or self-regulation, and ventromedial evaluation, the degree of functional connectivity between the two predicted successful weight loss. So all of this is consistent with the idea that self-control inhibitory systems, sometimes referred to as executive systems, are implicated in control of body weight. So in this study, we were interested to see if we could look for brain correlates of BMI. This is from the Human Connectome Project, which has actually 1,200 participants, healthy people in their 20s and 30s, who had very extensive imaging and behavioral evaluations. And we looked for correlates of BMI both in the cognitive domain, in the personality domain, and in the brain or cortical and subcortical volume domains. And the findings are as follows. First of all, we see that executive function, here as measured by cognitive flexibility, correlates with BMI. So better executive function, lower BMI. Interestingly, we also found a correlation with memory and spatial orientation processing, so visuospatial processing, and these are somewhat surprising findings. Another interesting finding, a negative finding here, is that there was no relationship with personality. And there's an immense literature relating, the BIG-5 personality measures to BMI, but we were unable to replicate it, although the N is only 900 in this case. But in particular, you see that there's no correlation between neuroticism and extroversion and BMI, and these have been linked to BMI in the past. We do find correlations between BMI and brain morphometry, and the interesting finding here is that we find that frontal asymmetry seems to be related to obesity. Namely, people with obesity seem to have thicker left frontal cortex and thinner right frontal cortex, especially this inferior frontal gyrus here. And this right inferior frontal gyrus has been implicated consistently in inhibitory control. The other thing that's interesting is that this left-right asymmetry was proposed to be related to impulsivity and to obesity based on lesion studies over 10 years ago. So it may be that it is not just frontal morphometry, but frontal asymmetry that relates to the phenotype of uncontrolled eating. This needs to be replicated, however. Now, the HCP has twins. It has both mono and dizygotic twins, allowing us to measure heritability. And what you can see is that BMI has a heritability of 70% in this sample, which is very consistent with the literature. But then we looked at the cognitive, personality, and brain morphometry traits associated with BMI, and we see that they're also very heritable. In other words, not only is BMI heritable, not only is it related to brain and cognition, but the cognitive and brain features related to obesity are themselves heritable. Now, the relationship between BMI and impulsivity, which as I showed you we could not replicate, but has been reported consistently, may be in a way an artifact, and it may be an artifact of the scales. So this is the N5 impulsiveness facet from the BIG-5, and Uku showed that its relationship with BMI was entirely driven by two questions. Namely, I tend to eat too much of my favorite food, and sometimes I am not able to control my appetite. In other words, people with obesity are not impulsive. They just answer yes to questions related to food. And this may not be a reflection of impulsivity. It may just be a reflection of being overweight and trying to lose weight, and basically experiencing this feeling of not being able to control your appetite. So Uku and I then looked at this idea of food addiction so what Uku did, he did meta-analysis of all the studies that related the BIG-5, this personality questionnaire, which has these 5 traits, and each of which has multiple facets. And he looked at their relationship with obesity, but also the relationship with addiction, in particular alcohol, gambling, opiates, and smoking addiction in this case. And what you can see, so two of the commonest are neuroticism and conscientiousness. And you can see that, there's a strong association for the addictive drugs. But for obesity, the association is much less and may not even be present. So it suggests that the concept of food addiction may be incorrect, at least as relates to personality profiles related to addiction. And then he related obesity to the other addictions via these domains and facets. And what you can see is that when you look at facets, obesity really falls away from the addictions. So all of the addictions overlap in terms of personality dimensions, but obesity seems to be apart from it, suggesting that maybe the concept of food addiction needs to be viewed with caution, at least from the personality standpoint. So here is more puzzling evidence regarding the link between self-control and obesity. So I've told you that there's this consistent relationship in the literature between executive function and BMI, and I've shown you maybe why that would be the case. But this study is kind of interesting. So here, it's quite recent publication. They have people on a drug called semaglutide, which is a GLP-1 agonist, and it's a drug. It's also called Wegovy, Ozempic. It's a drug that is associated with very significant weight loss. And the reason it causes weight loss is that GLP-1 is a satiety signal. It's secreted by the gut at the end of a meal to stop eating. And so when you give a GLP-1 agonist to people, they are just less hungry. And here's the study. You can see that versus placebo, there's very significant weight loss on semaglutide, 16% weight loss. But then this group administered a food craving questionnaire. So a questionnaire that's sort of in the domain of food addiction. And they found that semaglutide reduced scores on this craving questionnaires. So reduced craving for savory and sweet foods and increased craving control. And craving control is just a questionnaire measure, very similar to BIG-5, but specifically food related. It asks people, how good are you at controlling your food intake? And it's not surprising, I guess, but what they show is that reducing hunger with a hormone, and there's no reason to believe that GLP-1 agonists affect frontal function or executive function, but reducing hunger increases control of craving. And so it's quite possible that all the findings so far on executive control and feeding are very strongly driven by the experience of the person. If you are not overweight, you will not experience difficulty controlling your food intake because it will be automatic and not hard for you. Whereas if you're overweight and constantly going on diets, when someone gives you a questionnaire about self-regulation, you're more likely to answer that it is something that you have difficulty with. The other puzzling piece of evidence is also a recent paper. I showed you before that obesity was genetic and polygenic. So here we see this relationship between the polygenic risk for BMI and BMI is very strong. But in this paper, they show that it's mediated by diet quality. And here diet quality has to do with whether you eat processed foods, junk food, or not. So this is kind of interesting. So people at genetically high risk for weight gain tend to consume an unhealthy diet, which brings back this notion of executive control, decision-making, economic decision-making. So just to show that the evidence is not 100% clear. So I've shown you that there's pretty consistent evidence of cortical and subcortical measures being related to BMI. So that does support the view that cognitive control may play a role in obesity. However, what if the relationship between brain and BMI was not that the phenotype of uncontrolled eating was related to brain, but that adiposity itself was related to brain morphometry? So we know that obesity is a risk factor for Alzheimer's disease and dementia. Indeed, it's the number one modifiable risk factor for dementia and Alzheimer's disease. And it's been estimated recently that if you could correct the BMI, so people who are obese, if you could reduce their excess BMI by 50%, you could reduce their risk of excess mortality by close to 50%. Here in this meta-analysis, the authors looked for all of the factors that change the relative risk of Alzheimer's disease. So this is just Alzheimer's disease. And what you can see, it's difficult to read, but so many of these factors have to do with cerebrovascular disease, cerebral microbleeds, low cerebral perfusion measured by MRI or PET, some evidence of metabolic disease like having had bypass surgery or having had a stroke, having diabetes. And you can also see that obesity in midlife is associated with an increased risk of Alzheimer's disease. So in other words, many factors associated with an increased risk of dementia are related either to obesity or to the complications of obesity. So with Philip Morris and Masa Dagher, we looked at the relationship between adiposity and brain in the UK Biobank. So here we're looking at 20,000 individuals, mean age of 63. So we have, this is a structural equation model, and so we have obesity, which is measured by waist to hip ratio, BMI, and body fat percentage. And then we have these four latent factors which represent the metabolic syndrome, namely hypertension, diabetes and insulin resistance, inflammation, measured by blood markers, and dyslipidemia, also measured by blood tests. And what you can see is that obesity is related to these three metabolic derangements, and these are themselves related to cortical thickness, cortical volume, subcortical volume, and the presence of cerebrovascular lesions in white matter. And these, in turn, are associated with impaired cognition and reduced cortical volumes. So, in other words, what this shows is that adiposity is related to cerebrovascular risk factors, which is then related to brain atrophy, and then related to cognitive decline. We then looked at the pattern of cortical atrophy. So many studies have shown that obesity in midlife is associated with loss of cortical volume. But here what Philip did was to try to relate the spatial pattern of adiposity-related cortical thinning to Alzheimer's disease. And what he found is that when you look at AD patients and compare their maps, so this is a brain map of tissue loss in Alzheimer's disease, this is a brain map of tissue loss in obesity, you can see that there's very strong spatial correspondence, and this is using spin tests to correct for autocorrelation. You can see the same effect if you restrict your sample to lean AD individuals. So even AD patients who do not have obesity show a pattern of cortical thinning that is similar to the cortical thinning pattern in obesity. And as I said before, the relationship between adiposity and cortical atrophy is mediated by metabolic dysfunction, and these are among the greatest risk factors for Alzheimer's disease, but also for dementia in general and cerebrovascular disease. The point here is that any study that shows an association between brain morphometry and BMI, the direction of the influence is not obvious. So one way to get around this is to look at children. So I have about 5 or so more minutes. So This is data from the ABCD study, which is 9 and 10-year-old children in the US. And what you can see is that this is a paper that came out a couple of years ago by Ronan et al. And Paul Fletcher's group. And they show, first of all, that obesity in this group is related to executive function. So the same story, that the children with higher BMI have lower executive function. They also found that body mass index was related to reduced cortical thickness in the frontal lobes, as we've shown before in adults. And then they also found that there was a correlation between reduced executive function and cortical thinning. So the story it's kind of interesting and it's a nice story. Children with higher BMI have cortical thinning and reduced executive function, which may then lead to overconsumption. Now, there are things about this that to me don't make sense. So the first is that in childhood, cortical thinning is not associated with frontal lobe dysfunction. In fact, quite the contrary. This really quite famous study by Bethlehem et al., which looked at brain morphometry from birth to 100 years, shows that, first of all, cortical thickness varies in childhood. Let me see, cortical thickness is this one here. And what you can see is that during childhood, it is normal for there to be a loss of cortical thickness or cortical thinning. And in fact, this cortical thinning is associated with improving frontal lobe function as the child goes into adolescence. So in other words, in this study here, but there are many other studies that show this, cortical thinning is actually beneficial and associated with better frontal lobe function. So the previous study that I just showed you doesn't make any sense because it seems to relate cortical thinning to both worsened cognition and increased BMI. So I think something doesn't make sense about that finding. And one reason may be that there are many confounds. In particular, and this is a big problem in ABCD, is socioeconomic status. We know that socioeconomic status is associated with BMI, cortical morphometry, and cognitive function. And in older people, it's also associated with ill health. So that's a frequent confound in all of these studies that you must take into account. So we did a similar study in the ABCD, but instead of looking at BMI, we looked at BMI polygenic risk score. So we find, just like in the LOCK study that I showed before, that the obesity risk alleles are normally distributed in the population, and that the more alleles you have, the greater your BMI. So already at age 9, this polygenic risk for obesity manifests. And this, to me, reduces the likelihood that this genetic risk is via executive control. We don't know for sure, but it's likely that in 9 and 10-year-olds, food intake is much more driven by environment and by the parents than it is by your ability to control your appetite. But I don't know for sure. However, one problem that we found, so ABCD has 22 different scan sites, and what you can see is that BMI varies tremendously by scan site, and the BMI polygenic risk score varies by scan site. In other words, people who live in different parts of the United States have different genetic risks for obesity. And so you must correct for that. And you have to correct for it genetically. You have to remove the principal components of the GWAS to correct for this distribution. And the reason you have to correct for it, of course, is that these individuals are all scanned on different scanners, different makers, different cameras, different everything. So that will affect, any measurement that you make. However, what we did find in the ABCD study is that the body mass index polygenic risk score was associated, going in the same direction as the Ronan et al. Study, with reduced cortical thickness and reduced executive function. And then we did this Watershed model where we showed that BMI polygenic risk score was associated with lower cortical volume, which was itself associated with changes in self-control and impulsivity, here measured by urgency, which was then associated with BMI. I'm saying all this to say that I don't know the answer. I don't know if this is true. This is a small sample, relatively small. I mean, it's 3,000 people with multiple potential confounds. Finally, I mentioned that socioeconomic status was related to BMI. So in this paper with Herman Kwon and other collaborators, we looked at socioeconomic status in the UK Biobank, and we showed that socioeconomic status was associated with cortical thinning. This is well known. But what we also found is that 40% of the non-heritable variance in socioeconomic status was mediated by BMI. So these findings are all a little bit confusing, and I think that we need to do more work to try to understand them. But I think we can say that there is an obesity endophenotype, which we can call uncontrolled eating. It can be identified by personality questionnaires, but the findings are inconsistent. But what people have published is that reduced conscientiousness or self-regulation appears to be associated with weight gain, and there may be a relationship to more impulsivity measures such as neuroticism and extroversion. However, the relationship there is much less clear. Reduced executive function may be heritable and may already be present very early in life, and it may be that these brain systems are not only involved in executive function and self-control, but also involved in the regulation of energy homeostasis. The rising incidence of obesity is probably related to food abundance and to cheap food, and I propose this idea that economic optimization means that it is optimum to overeat when food is abundant, and that maybe body weight defense is under control of both homeostasis, peptides like ghrelin and leptin, but also cognitive control. And finally, I showed that adiposity itself affects the brain. Midlife adiposity is associated with cortical atrophy. It's the #1 modifiable risk factor for AD and dementia, an association that's mediated by hypertension, hyperlipidemia, insulin resistance, and inflammation. And I think that treating obesity with novel medications like semaglutide will actually be seen as very important for the prevention of neurological complications. So I will end there, and I'm happy to take questions.
Thank you. Thank you so much for this really interesting talk. So as you just now said, we have now time for a few questions. So please feel free to post them now in the Ask the Questions button, and I will read them out. And also don't forget that after the talk we will meet for a short meet the speaker session, so you can also ask more questions there. And I will start with one question that asked about what you just mentioned, the novel treatment therapy semaglutide, so the GLP-1 agonist. So the comment is, thank you for the interesting talk. In the study in which they presented the GLP-1 agonist, do you expect the activity on those brain regions you have identified before to be altered? Meaning, can that hormonal treatment alter executive function?
Yeah, so in that study I showed the hormonal treatment affected executive function as measured by questionnaires. But it would be extremely interesting to see with, let's say, fMRI studies. Oh, sorry, there's an echo. Okay. Yeah, it would be very interesting to see with fMRI studies whether drugs that are GLP-1 agonists change self-regulation signal with respect to foods, but also with respect to non-foods. There's also actually evidence that semaglutide reduces smoking and alcohol intake. So yeah, interesting studies to do.
Thanks for answering, and I also will mute you while I read out the questions to remove the echo. There is a different question about, also first thanking you for the interesting talk, and if you looked at any sex differences in the correlates for obesity, or if you know about any sex differences.
Thanks. Actually, there are sex differences in almost every aspect of obesity, going all the way from biological to social and psychological, and there would be at that level gender differences even. And this is something that is, and there's also differences in the incidence of dementia and Alzheimer's disease. It's more common in women. And there's differences in the incidence of cerebrovascular disease, hypertension, and metabolic syndrome. They're more common in males. So there are multiple sex differences, but as far as sex differences in the brain, we have not had the power, I think, so far to look at those. So we have not. And I'm not aware of anyone who has.
I think it's a topic that is becoming more and more prominent these days, so I hope in the next few years we'll do more research on sex differences. One of the questions is also if there's any data regarding the brain lipid composition in obese versus lean individuals.
That's a good question. So the question relates to lipid composition. That I don't know about. I do know about white matter changes. So we do see, I didn't show this, but using diffusion imaging, we also see very prominent changes in white matter and myelination related to executive function and to BMI. But does adiposity change the brain constituents, lipid constituents? That I don't know. What we do see, however, which is quite puzzling in a way, is people who have bariatric surgery. So bariatric surgery causes very significant weight loss and changes in metabolic derangements that are very rapid. And we see very immense changes in cortical and white matter measures, basically going in the correct direction, going in the direction of health, brain health. And we see those even at 4 months, and I don't know what is causing those. So for example, changes in myelination as measured from T1 MRI, as soon as 4 months after bariatric surgery. I can't believe that it's due to myelin formation, but it could be related to lipids.
Thank you. Actually, a follow-up question. You said after 4 months, are they also permanent then? Are there any follow-ups?
Well, up to 24 months. And there's consistent amelioration in brain health parameters after weight loss. But most of the studies, ours and other people's, are sort of convenience samples where people use T1 MRI or sometimes diffusion imaging, but it would be very interesting to use more advanced measures of cerebrovascular health after weight loss to see how to see what is happening in the brain.
Thank you. Just taking a second because I'm muting and unmuting and switching. There's another question, again, thanking you for the talk and saying that BMI is related to an earlier puberty in children. So could it be that in childhood an increased thinning caused by obesity is related to this progression?
That is a very interesting theory. In the ABCD study, I don't think we have good measurements, although there might be. So we have not looked at it, but it is really a very interesting question. So very low BMI and very high BMI are both related to endocrine changes in childhood. So this is something that we should look at as well. Thank you.
Thank you. And I think a last question that I'm seeing here, are there correlations between conditions with executive function impairment, autism spectrum disorder, or ADHD, for example, and obesity?
Yes, there are. So ADHD is associated with obesity. It's also associated with executive dysfunction. And in fact, if you look at people who have severe obesity and who go for bariatric surgery, they either have ADHD or score very high on measures of attention and cognitive control. So there's a link, and there's a genetic overlap. There's a genetic overlap between ADHD and BMI. So there's a genetic correlation between the two. And there's a genetic correlation with another condition called binge eating disorder, which also overlaps significantly with ADHD and interestingly responds to the same drug treatments. So, and there's a recent paper, I don't remember the reference, I think it's a preprint maybe, that tried to do genetic analysis of the overlap between ADHD and BMI. I saw it on Twitter this week, but I don't remember where it is.
Thank you. Yes, very interesting. The high overlap between ADHD and obesity, and also that there was recently this paper, or generally speaking, showing the same medication is basically helpful in binge eating, even though we don't exactly know how it works. There are also other comorbidities with obesity, like depression. Do you think this is a similar link, or you think this is for some other reason?
I think I think it is a similar. There is overlap. So I didn't talk much about inflammation, but we think that one of the main ways adiposity changes the brain is via inflammatory markers. These are also implicated in depression, and they're also implicated in depression associated with BMI. There's also a social factor relating obesity to, mood disorders. So it's a very complicated interaction. Again, something that I think to study this, we need big data like the UK Biobank, and it's really interesting that we're more and more able to study these things.
Okay, thank you very much. We're also very good in time, so if there are no other questions in the chat right now, don't think so, I would suggest that we can head over to Zoom for some more informal questions maybe. So I will post the Zoom link now in the chat. It's here, so everyone please feel free to join. And other than that, thank you again, Alain, for this really interesting talk and summarizing so many findings from different parts that first don't seem to be related that much, but actually it seems to create a whole picture that makes sense. And thank you, the audience, for joining us today. And next week we will host Emily Jacobs. She will talk about dynamic endocrine modulation of the nervous system. So hope to see you there as well, and hope to see many of you now on Zoom.